So we left off in class on Thursday talking about the different types of microscopy. So really briefly, I'm going to talk about how to prepare specimens for microscopy and also different types of preparations that one could do in a microbiology lab. And again, I know this is going to be a review for what a lot of you are doing in the lab or have done if you've already taken the lab. But for those of you who maybe are not taking the lab, this will be important information that's relevant to the topic of the course. So we have to, especially when we're looking at bacteria, bacteria are very difficult to see because they don't contrast very much with the background. So we have to stain them or do something to them to allow us to actually see their morphology or to see any sort of structures. So staining is the process by which we use a dye to falsely color the microbe. The microbe isn't actually purple, but if we use a purple dye, we will see a purple bacterium. We can also use various staining techniques to emphasize certain structures. So one of the first steps that we'll do, and that you will do in the lab when you are going to prepare a slide to look at, is you take a smear, a thin film of bacteria usually, and place it on the slide. And this can come either from a plate or a liquid culture, and in lab you'll learn how to do that. You then have to do what's called fixation. If you just start trying to stain and your cells are just like right there on top of your slide, a lot of them will fall off during the wash steps. So by doing this process called fixation, you basically are sticking them to the slide so they can't fall off when you start to do the staining process. You can use heat, sometimes you can use alcohol. In the lab here we use heat. So you pass it briefly through a Bunsen burner, through a flame, in order to cause the cells to adhere to the slide. We do it briefly because you can also cause them to explode if you boil them. So the basic principle behind how staining works is that stains can either have a positive or a negative ion. If you have what's called a basic dye, the color, the dye, is a cation. It's a positively charged ion. Some examples that you might use in the lab include crystal violet, methylene blue, and safranin. So the principle behind this is that because the dye is a positively charged ion, it will be attracted to and stick to the negatively charged cell of the bacterium. The other type of dye that you might use in the lab, at least once, is an acidic dye. So in an acidic dye, the dye molecule, the chromophore, is an anion. It's negatively charged, a negatively charged ion. And again, you might kind of see where I'm going with this. If the bacterium is negatively charged and the ion is also negatively charged, in this case, the bacterium actually repels the dye. So you stain the background instead of staining the bacterium itself. We can also use things called mordants. These are reagents that allow us to hold the stain. And when we talk about a gram stain in just a minute, I'll tell you what the mordant does there. You can also use a mordant to coat small structures like flagella so they're easier to see. And the purpose of doing a stain is just so that we can look at the shape, the arrangement, and the basic structure of microorganisms. So in this image here, these are basic dyes. So they have a positive dye molecule. They're attracted to the negatively charged cell. Here's crystal violet, they're purple. Here's methylene blue, they're blue. Here's safranin, they're pink or red. So you don't see much besides the shape of the cell. These look like rods, arrangements, they're kind of in chains. And we don't really see any basic structures because these are pretty small. This is an example of doing an acidic dye where the chromatophore or the dye molecule is negatively charged, so it's repelled by the cell. And you can actually, I like negative stains. They're a little bit tricky to do, but I really like them when they work well. So these are also rod-shaped bacteria, but you can even start to see where they've like started to divide, and you can see the little cell divisions there. So it gives a little bit better detail of the surface of the cell. But sometimes we might want to be able to not only look at the cell, but see if they have some sort of different properties. And the most common stain that you're going to do in the lab, and I will tell you right now, those of you who are in the lab, your midterm lab practical, you will have to do this. It's not a secret. You will have to do it. You will be graded on your ability to do it, is a gram stain. And the gram stain looks at differences in the cell wall. The other type of stain that's a relatively common differential stain, you won't be doing it, but you should be looking at cells that have had this done, is an acid-fast stain. And that stains cells that have a different type of coating around their cell wall. So the gram stain is very common, very popular, very widely used. And it allows us to identify whether or not bacteria are gram-positive or gram-negative. So cells that we call gram-positive have a very thick layer of peptidoglycan. They have a very thick cell wall. And they have only a single membrane. Cells that we call gram-negative actually have two membranes. So they have like an inner plasma membrane and an outer membrane. And in the middle, they have a very thin layer of peptidoglycan. Now, this concept is actually really useful in a clinical setting. So I like to, when I teach classes like this, I like to tell you not only the technique and that you need to know the technique because it's going to be on your exam, but what is the relevance? Like, who cares if a cell is gram-positive or gram-negative? Well, if you're interested in healthcare at all, including your own, it's important because antibiotics affect these different types of bacteria differently. So gram-negative bacteria tend to be more resistant to antibiotics, especially things like penicillin that affect the cell wall, because they have such a little cell wall. They don't care if it's gone. They don't really need it. But gram-positive tend to be really sensitive to antibiotics like penicillin. So if your infection is gram-negative and your doctor's like, here you go, here's your penicillin, you're like, no, please give me something else. Okay, so it can be very, very important. The basic process for doing a gram stain is shown here. After you have smeared and fixed the cells, you add crystal violet. So remember, crystal violet is a simple stain that is positively charged. It's basic. So the positive ions of the crystal violet are attracted to the negatively charged cell. You wash away the excess crystal violet, then you add iodine. So iodine is called a mordant. It actually will bind to the crystal violet to help it stay in the cell wall a little bit better. Then you do a step called decolorization. So you use alcohol very quickly to rinse away crystal violet. You're like, why do I want to do that? Didn't you tell me I need to see the cells? Yes, I did. But this is where gram-negative and gram-positive cells behave differently. So what happens is that because gram-negative cells have such a thin cell wall, even though the crystal violet has the iodine and it's a bigger complex, that thin cell wall releases the crystal violet. But the thick cell wall of the gram-positive holds it in still. So they are still going to be purple. So at this step, and this is also important, so I'm going to even note this on the slide. So all cells here are purple. Right? All cells here are purple. After step 3, gram-positive cells are purple. Gram-negative cells are clear. And you can't see clear. Clear's hard to see. So we still have to do a stain. So we add a second stain called a counter stain, and it's a different color. In this case, we're using safranin, which is like a pink or red. And so after this step, what should happen is our gram-positive are still going to be purple because they retained that crystal violet, so they're not affected by the safranin. Our gram-negative are now red or pink. Okay. When I teach the lab, I literally ask a question like this on the written portion of the midterm. These are the steps of the gram stain. What color are the cells after each step? So I'm telling you right now, like if I were teaching your lab, I would ask you that question. Just saying. So this is what an actual image of a gram stain would look like, one that's properly done. You would say that these cells here, these purple cells are gram-positive coccus-shaped cells, circular, and then these pink ones, these elongated rods, gram-negative rod-shaped cells. The acid-fast stain is used to identify species belonging to the genus Mycobacterium and Nocardia. They have this substance on their cell walls called mycolic acid. It makes them very waxy, and what it does is it also makes them more resistant to a gram stain. The crystal violet can't really get in there. The safranin doesn't really do well at getting in there, and so they're not really affected by it. So in this case, there are three major steps. You do the primary stain, which is a stain called carbonyl fuchsin. It turns everything red. When you decolorize, you add the alcohol. Anything that has that waxy layer still is red, but if it doesn't have the waxy layer, it loses the color, so it's colorless. And then you counterstain with methylene blue. So the cells that have that waxy layer should still be red, whereas the cells that don't will be blue. And so that looks something like this, where you have other things in there. They could be eukaryotic cells, they could be something, but you actually can see the Mycobacterium tuberculosis. We have special stains as well that can allow us to look at parts of a bacterial cell. So for example, the capsule, the endospore, or the flagellum. So a capsule is a polysaccharide, kind of gooey, sticky coat that some cells produce. You can't stain it really. And so what you have to do is a little bit of both. You do a negative stain and a basic stain. So the negative stain stains the background so you can see the outline of the capsule. And then you use a basic stain, or just a positively charged ion, that will go in there and actually stain the bacterium, but the capsule still doesn't get stained. And so what you end up seeing is this halo, and the halo represents that gooey, sticky capsule on the outside. You can also stain for endospores. Some bacteria form these really resistant structures called endospores under harsh conditions. We saw that example on the first day of scientists that were using the hay infusion to try to determine spontaneous generation versus biogenesis. And the bacteria in the hay actually formed spores, so they kept growing. So these are tricky to stain. It's actually, we call it boiling the heck out of them. But what you have to do is you get a culture that should form stains. It's usually bacillus, and you add a stain called malachite green, and you do this under high heat. So you are boiling the cells, literally boiling them on the slide. So you keep adding the green so you have it liquid, and it just boils and boils for 5 minutes. Then you decolorize with water, and then you counterstain with safranin. And what you should see is, like in this image, the spores are all these little green structures, the endospores, right? And then anything that's red is a vegetative cell. So you will be doing this type of stain as well. And then finally, you have the flagellar stain that can allow you to actually see the flagellum. So you won't do this process in the lab, but you will be looking at slides that have been processed this way. And so again, the way this works is it uses a mordant to coat the flagellum and make them thicker so that they're easier to see under the microscope.